Okay, go ahead.
Thank you.
Yep.
Yeah. Hello, everyone. Good afternoon. I am Manoj Natarajan doing my master's in Dalhousie University and working as a abstract student at Agri and Agri-Food Canada under Dr. Kesav Singh. Today, I am going to present on topic developing a low-cost digital imaging system for plant phenotyping using Raspberry Pi computers. Moving on to introduction. Traditionally, phenotyping in plant breeding is time-consuming, intensive, and data collection is insufficient to process the data to satisfy the need of plant breeders, which hampered the breeding progress. In recent years, advanced in digital electronics and sensor technology in agriculture have aided in the development of innovative method for measuring plant phenotypic characteristics. In this study, the Raspberry Pi based automation system was integrated with the image sensor, Sony IMX219PQ CMOS 8MP camera in growth chamber to timely monitoring the crop growth parameters in wheat breeding trial for plant phenotyping. The Raspberry Pi system was programmed using open source Python scripting to capture high resolution multiple imaging in user defined time interval, which is approximately 4 minutes throughout the experimental cycle. From this image allow us to study about the morphological phenology such as structure, plant height, plant density and volume, canopy cover, leaf area index, anthesis timing, maturity and digital biomass, and other physiological parameter with respect overall plant growth and health. Moving on to materials and methods. In this study, the main hardware used were from Raspberry Pi module with the accessories are shown in the figure 1. So the Raspberry Pi module we used was 3B and 4 module, which has a quad-core 64-bit 2 GB processor to control the image capturing technique. The Raspberry Pi module were connected to the Raspberry Pi camera version 2 via the camera serial interface port on Raspberry Pi microprocessor. These camera are based on the image sensor Sony IMX, which is 8MP megapixel and the active pixel size of up to 3280 into 2464. To manage the lack of availability of Wi-Fi, we use the real-time clock that is used to determine the correct time of the image capture. And an electronic fan was also used in this system to control the heat dissipated from the Raspberry Pi as it's running continuously. And this Raspberry Pi module need a power where we used a 26800 mAh solar power battery which can be directly connected to AC supply or through the solar light. And software used here are the Linux-based operating system. Raspberry Pi Buster was run on the Raspberry Pi microprocessor with open source Python C software using utilized primary programming software language to command the board to capture a particular image at a certain time interval. So initially the flowchart was designed for doing the programming. The necessary libraries were installed and the time function were already included in the programming. So initially the program was automatically adjusted for white balance, maximum white imaging resolution, frame rate, exposure mode, shutter time, and which is associated with the greenhouse conditions. We programmed the camera for three image with a time delay of 3 seconds, which capture 5 image in a total time period. So for every day we capture this image with the 4 hours interval time. So finally we get 330 images per day. As the real-time clock of Pi 3 was synchronized with the Raspberry Pi system, the captured image was recorded and saved in the file name format as shown in the file format, which is a date, month, um, year, hour, minute, camera name, and the Raspberry Pi module we used. The camera system was scheduled to power on and off based on the program management, which help us to store the particular image on the time. The experimental setup we chose where for plant phenotyping was carried out on growth chamber at Lethbridge Research Center and Development Center at Alberta. as shown in the figure 3. The growth chamber has two sides where five shells were installed with the Raspberry Pi module to capture the digital image. Figure 4 represents the one side of the growth chamber and figure 5 represents the mounting view of the camera. So the Raspberry Pi and its peripheral device, which include the power bank and the peripheral device, are tied with the zip ties on the shelves so that it can stay firmly on the shelf to capture the image. So as shown in the figure 3, the camera was set at a height of 170 centimeter and it covers like a 16 plant per each shelf with a dimension of 127 into 127 centimeter. So from this stuff, the proposed research experimental setup is able to capture 30 image in a day. The Wi-Fi hotspot was used for data transferring from Pi to laptop. The image processing software and Python scripting were used to analyze the digital image captured by the Raspberry Pi camera with the following main important three steps for image processing. First one is object segmentation, which will be used to segment the plant region in the image capture as shown here. And the second one is like extracting the plant leaf in the image capture, which determine the how green it stabilizely shows the chlorophyll value and other morphological characters. And the third thing is analyzing the morphological analysis, which used to analyze and process the extracted plant pixel based on the shape, which is help to determine the plant structure and the plant growth. Moving on to conclusion, the Raspberry Pi system found promising to automatically capture the spatiotemporal images to identify the plant growth and structure. The images have been timely analyzed to extract the morphological feature and the physiological characteristics of the plant. These studies suggest that the developed low-cost digital imaging system can be integrated with the Internet of Things based on sensor system, which help the plant breeders to make a timely decision during their plant growth, or whether in winter season or timely in spring season. The reference are listed here. These are the reference I choose to create these posters. I would like to thank the author would like Agri and Agri-Food Canada for supporting the ABASE and Western Green Research Foundation project. We also acknowledges our lab group met with who has a winter serial group members for their supporting in setting up the experimental to conduct this study, and also the funding support from the following institution. Thank you.